Okay, so this is the second part of the recording, although it's the third recording. This is cardiac muscle tissue anatomy and electrophysiology. Okay, so electrophysiology, the heart does not require conscious intervention to elicit cardiac muscles to contract. Cardiac muscle exhibits autorhythmicity, which means that it sets its own rhythm without the need for input from the nervous system. So unlike skeletal muscles, where in order for you to actually get a contraction, you must have nervous system intervention, cardiac muscle cells will contract in response to electrical excitation in the form of action potentials. So essentially, you're still going to get those action potentials to send the message to the cardiac muscle cells. It's just that they're not going to necessarily occur by intervention of the nervous system, which is what has to happen for skeletal muscles and smooth muscles. The cardiac electrical activity is coordinated by a very small, unique population of cardiac muscle cells called pacemaker cells. These cells rhythmically and spontaneously generate action potentials. They spread to the other type of cardiac muscle cells, such as a domino effect then, which are referred to as the contractile cells. Cardiac muscle cells, like skeletal muscle fibers, have striations, alternating light and dark bands when viewed under a microscope. As with skeletal muscle fibers, striations are due to the arrangement of contractile proteins within the cardiac muscle cells. Structural similarities reflect the physiological similarities. Skeletal and cardiac muscle tissues have the same general functions. Both generate a tension through a sliding filament mechanism of contraction. Cardiac muscle cells are typically branch cells with a single nucleus. They are shorter and wider than skeletal muscle fibers. They also contain an abundant myoglobin. Myoglobin is a protein that carries oxygen. It is different from hemoglobin, and most significantly, it is different in that hemoglobin, as we talked about with the red blood cells, carries 4 oxygen molecules. Myoglobin will actually only carry 1 oxygen. Nearly half of cytoplasmic volume is composed of mitochondria. This is really to reflect the high energy demands, as the mitochondria is going to be responsible for producing that ATP that's going to be required for these contractions to occur. The cardiac muscle cells will possess unique structures called intercalated discs that join adjacent cardiac muscle cells. They will also join pacemaker cells to contractile cells and contractile cells to one another. The intercalated disc will contain desmosomes, which will hold cardiac muscle cells together, and gap junctions that will allow ions to rapidly pass from one cell to another, permitting communication among cardiac muscle cells. And here you can see, looking, this is the general structure of the cardiac muscle. Looks somewhat similar to that of skeletal muscle, but there are going to be those differences, mainly those gap junctions. Like skeletal muscle fibers and other excitable cells, cardiac muscle cells contain a selective gated ion channels in the sarcolemma. Opening and closing action of these ion channels is responsible for both pacemaker and contractile cardiac action potentials. There are several different types of gated ion channels. We have our voltage-gated sodium ion channels, open and close in response to a change in voltage. In all cardiac muscle cells, except certain pacemaker cells, you will find these voltage-gated sodium ion channels. The calcium ion channels, these demonstrate voltage-gated opening, but time-gated closing. They will close after a certain period, regardless of the voltage. All types of cardiac muscle cells have one or more types of potassium ion channels, and some are ligand and others are voltage gated. Now, the pacemaker cells undergo rhythmic spontaneous depolarizations that lead to the action potentials. Once you do fire that action potential, it spreads quickly through the heart by the cardiac conduction system. This is a group of interconnected pacemaker cells. The action potentials are transmitted from the pacemaker cells to the contractile cells through the intercalated disc. Gap junctions in these discs allow electrical activity generated by the pacemaker cells to rapidly spread to all cardiac muscle cells via the electrical synapses. Again, they're connected by these electrical synapses, so you really get that full domino effect. This will permit the heart to contract as a unit and produce coordinated heartbeat. Reason cells of the heart sometimes referred to as a functional syncytium, meaning that all the cells will contract as one unit. The pacemaker cells make up only about 1% of the total number of cardiac muscle cells. 3 populations of these cells are located in the heart, capable of spontaneously generating action potentials, thereby setting the pace of the heart. 3 cell populations are collectively referred to as the cardiac conduction system. So you're also going to establish what we refer to as a pacemaker potential. This is much different from that of the contractile cell. Depolarization in a pacemaker cell occurs much slowly due to the lack of voltage-gated sodium ion channels. The pacemaker cell action potentials lack a plateau phase, and membrane potential oscillates, never remains at resting level. Instead, it occurs in a cycle, with the last event triggering first. This occurs because nonspecific cation channels. These are unique to the pacemaker cells. And what happens, essentially what's happening here, let me see if I actually have a little bit of a picture, is in these particular cells, you have these ion channels. They're leaky channels. They're always open. And what happens is they're slowly leaking in positive ions. And what happens is you're slowly going to eventually reach that point of threshold in which you fire the action potential, which then triggers the whole domino effect. So the reason why these are pacemaker cells is because they have these leaky channels that, instead of staying at resting membrane potential and maintaining resting membrane potential, they are always allowing for an influx of positive ions, which will automatically get them to that threshold in a rhythmic pattern. And again, once you repolarize here, you can see that this whole cycle starts over again because of these leaky channels. Okay. So there are 3 populations of pacemaker cells. The first is the sinoatrial node, or SA node. This is in the upper right atrium, slightly inferior and lateral to the opening of the superior vena cava. Under normal conditions, the SA node has the fastest intrinsic rate of depolarization, that of about 60 or more times per minute. The rate is subject to influence from sympathetic and parasympathetic nervous system. So your autonomic nervous system can come in and modulate the rate of the contractions of the heart, but it's sort of set by these pacemaker cells. You then have the atrioventricular node. This is posterior and medial to the tricuspid valve. It is a bit slower than the SA node, the intrinsic rate of only about 40 action potentials per minute. This is sort of a backup system. You know, if your atria don't actually contract, that's not necessarily the end of the world, because 80% of blood still moves passively through. But if the ventricles do not contract, that is serious, because now you're not actually pumping blood through the pulmonary and systemic circuits. So this is a backup system. If the SA node fires, it will trigger the firing of the AV node. If the SA node fails to fire, that will then allow for the AV node to eventually kick in and fire. That's why it's at a slower pace. The Purkinje fiber system, this is the slowest group of pacemaker cells, depolarize only about 20 times per minute. Atypical pacemakers, because their action potentials rely on different ion channels, and they actually function a little bit different. The Purkinje fiber system includes the atrioventricular bundle, the right and left bundle branches, and then their terminal branches. So generally what happens is the SA node would fire. That would then trigger the atria to contract. When the atria contract, that will then trigger the AV node to fire, which will then trigger the ventricles to contract. If the SA node fails to fire, eventually you will not get contraction of the atria, but what will happen is 80% of blood will passively move in. But at some point, hopefully the AV node fires because it's at a slower pace, which means the ventricles would still fire. You'd still be able to move blood through pulmonary and systemic circuits. Sinus rhythm, each population of pacemaker cells can potentially pace the heart, make it beat at a certain rate. One that depolarizes fastest sets heart rate. The other pacemakers pace the heart only if the fastest pacemaker ceases to function. So, the SA node is the normal pacemaker of the entire heart. Electrical rhythms generated and maintained by the SA node are known as sinus rhythms. The AV node and the Purkinje fiber system normally only conduct action potentials generated by the SA node. If the SA node ceases to function, the AV node can successfully pace the heart, albeit at a slower pace. The AV bundle of the Purkinje system is only connection between the AV node and the ventricles. If blocked, the SA node cannot pace the ventricles, even if functioning normally. The Purkinje fiber system is capable of pacing the heart, but its slow rate of depolarization is not adequate to sustain life beyond a short period of time. Occasionally, a group of regular contractile cells or pacemaker cells, other than the SA node, will attempt to pace the heart at the same time as the SA node, such as extra pacemaker, is called an ectopic pacemaker, and this can cause irregular heart rhythms. Under normal conditions, the SA node generally generates action potential. This spreads rapidly via gap junctions to the surrounding atrial cells. Impulses are conducted by specialized atria conducting the fibers to the AV node. Altogether, this requires about 0.03 seconds. Once the impulse reaches the AV node, the conduction slows considerably as a result of the low number of gap junctions between the AV nodal cells and the presence of non-conducting fibrous skeleton that surrounds the AV node. This slow conduction, which is referred to as the AV node delay, generally lasts for about 0.13 seconds. This allows the atria to depolarize and contract before ventricles, gives the ventricles time to fill with blood. It also helps to prevent the current from flowing backward from the AV bundle into the AV node and the atria. The action potential is then conducted from the AV bundle to the right and left bundle branches. Depolarization spreads along the Purkinje fibers to the contractile cardiac muscle cells of the ventricles. The contractile cells, the great majority of the cells that make up the cardiac muscle cells, action potential in the contractile muscle can result from reversal of membrane potential, just like we see with skeletal muscle. In this case, the voltage-gated ion channels in the sarcolemma and unequal concentration of sodium and potassium ions on either side of the membrane drive ions in or out of the cell through these channels. The sequence of events in the contractile cells is you have a rapid depolarization phase, initial repolarization phase, then you have a plateau phase where the depolarization is sustained at about 0 millivolts in the plateau phase. This is a critically important phase. It is really due to the slow opening of the calcium channels. And then you have the repolarization phase. This is the final phase where you repolarize. The plateau phase is really a little bit of an extended phase there to allow for proper ejection of the blood from the ventricles. So here you can see the rapid depolarization. Unlike with the skeletal muscles, you go straight up almost immediately. You have a little bit of an initial repolarization, then you have this plateau phase where you don't really see much of a change, and then you have the repolarization phase where you come back down to resting membrane potential. The sequence of events of the contractile cell action potentials resembles that of the skeletal muscle fiber action potential, with one important exception, that of the plateau phase. If cardiac action potentials lasted only about 1 to 5 milliseconds, like skeletal muscle fiber action potentials, the resting heart rate would be about 15 times faster than it should be at rest. The plateau phase actually lengthens the cardiac action potential to about 200 to 300 milliseconds. This slows the heart rate, provides time required for the heart to fill with blood. Okay, and this gives us what we refer to as an effective refractory period. Again, allowing that time for that heart to sort of fill up there. The plateau phase also increases the strength of the heart's contraction. Prolonged action potential makes the muscle twitch less longer and can develop more force. This allows more calcium ions to enter the cell, which is needed for the contraction via the sliding filament mechanism. The plateau phase also effectively prevents tetany. This is a sustained contraction in the heart by lengthening of the refractory period. The refractory period in the cardiac muscle cells is so long that the cells cannot maintain sustained contraction. This allows the heart to relax and the ventricles to refill with the blood before the cardiac muscle cells are stimulated by an entirely new contraction. The mechanism for cardiac muscle cell contraction is very similar to that of skeletal muscle fiber. This occurs by the sliding filament mechanism, where we see the excitation, contraction, coupling. We saw this with the skeletal muscles. You're going to see the same thing here. Okay. So the process of the excitation, coupling, reaction is in a stimulated cardiac muscle cell. The depolarization will propagate through the sarcolemma and dives into the cell along a T-tubule. This causes the sarcoplasmic reticulum to release calcium ions. The ions will then bind to troponin, allowing actin and myosin to bind and the cross-bridge cycle to begin. The sarcoplasmic reticulum of the cardiac muscle cells is much less extensive than that of the skeletal muscle fibers, and does not release enough calcium ions to produce reliably strong contraction. The remaining calcium ions needed for the contraction will diffuse into the cell during the action potential through the calcium ion channels from the extracellular fluid in the T-tubules. For this reason, the concentration of calcium ions in the cardiac extracellular fluid plays a significant role in determining the strength of the contraction. This is why calcium ion homeostasis is so critical. And this is also, if you remember, if they're trying to start somebody's heart, they might inject calcium right into that extracellular space there. Looking at an ECG or an EKG, this is an important clinical tool for examining the health of a heart. It is a graphic depiction of electrical activity occurring in all cardiac muscle cells over a period of time. The recording occurs by placing electrodes on the surface of the patient's skin, 6 on the chest, 2 on each extremity. Electrical changes are shown on the EKG as deflections or waves. This will show changes in the electrical activity. If there is no net difference, there is no deflection shown. One of the most obvious changes in the heart revealed by an EKG is the disturbance in electrical rhythm. EKG recordings generally consist of 5 waves. Each represents active depolarization or repolarization of different parts of the heart. You have a small initial P wave, which represents depolarization of the cells within the atria except the SA node. The P wave nearly always registers as an upward deflection on an EKG. You have a large QRS complex, which represents the ventricular depolarization. Actually, three separate waves. Q is the first downward reflection, R is the upward deflection, and S is a downward deflection. They'll then have a small T wave, which occurs after the S wave of the QRS complex. This represents ventricular repolarization. The T wave is upward deflection under normal conditions. So here we can see atrial depolarization. This is contraction of the atria. That's your P wave. Ventricular depolarization. This is actually the QRS. This is a much more significant event as far as the electrical activity of the heart is concerned. You are repolarizing the atria, but unfortunately that process is masked because of the ventricular depolarization event. And then this is followed by the T wave, which is ventricular repolarization. Periods between the waves represent important phases of action potentials and the spread of electrical activity through the heart. The intervals include component of at least one wave, and the segments do not include any wave components. The three intervals are the RR interval. This is a time between two successful R waves. The entire duration and generation and spread of action potential through the heart. It can be measured to determine the heart rate. The PR interval, this is the period from beginning of the P wave to the beginning of the R wave. This is the time it takes a depolarization generated by the SA node to spread through the atria to the ventricles, including the AV node delay. And then finally, the QT interval. This is the time from the beginning of the QRS complex to the end of the T wave. Action potential spread through the ventricular cells. And again, here you can see the R interval, the PR interval, and the QT interval here. The ST segment, this is between the end of the S wave and the beginning of the T wave. This is flat because it is recorded during the plateau phase of the ventricles. No net changes occur in the electrical activity. Elevation or depression of the ST segment is seen with many clinical conditions, most notably myocardial ischemia and myocardial injury and infarction. And here you can see the ST segment there. So this is a typical EKG or a normal EKG, and you can see all outlined segments there. Cardiac dysrhythmias, these have three basic patterns. Disturbances in heart rate, you have bradycardia. This is a heart rate under 60 beats per minute. Tachycardia is a heart rate of over 100 beats per minute. Sinus tachycardia is a regular fast rhythm. Disturbances in the conduction pathways, normal conduction pathway that might be disrupted by the accessory pathways between the atria and the ventricles or blockage along the conduction system is referred to as a heart block. Blockage is common at the AV node. The PR interval is longer than normal due to increased time for impulse to spread to the ventricles through the AV node. The extra P wave present indicates some action potential from the SA node are not conducted through the AV node at all. Disturbances in the AV node blockage. Another common location for a heart block is along the right and left bundle branch, and this will generally widen the QRS complex. So essentially, if you look here, you're sort of missing one of the QRS complexes here. You have the PQRS T wave, P wave, nothing, and then another P wave. This is a heart block of the AV node. In fibrillation, electrical activity of the heart is essentially goes haywire, causing parts of the heart to depolarize and contract while others are repolarizing and not contracting. Fibrillating muscle is often visually compared to writhing movement of a plastic bag full of earthworms. So here you have atrial fibrillation, generally not life-threatening because atria contraction isn't necessarily from ventricular filling, manifests on the EKG tracing as irregular rhythm where you have no discernible pattern and it lacks P waves, as you can see here with some of these. Ventricular fibrillation is just a mess. This is an immediate life-threatening and manifests on the EKG as a chaotic activity. This is treated with fibrillation and an electrical shock to the heart, which will depolarize all ventricular muscle cells simultaneously and throw the cells back into the refractory periods. And then hopefully they'll be able to re-pick up with normal sinus rhythm. Ventricular fibrillation is not the same thing as flatlining, a condition known as asystole. Defibrillation is not used for asystole because heart is not fibrillating. No electrical activity to reset. Treating with CPR and pharmacological agents that stimulate the heart, such as atropine or epinephrine, is typically something that's used. Okay, and that is the end of part 2 of the heart.